Thank you, Prapan, and it's always a great pleasure to be here in January at the Bangkok Symposium, and I think the relevance and the quality of the presentations, at least up till now, is a great tribute to the organising committee and the hard work they put in. So I'm going to restrict my talk today to HIV vaccines. I think Eugene dealt well with immunotherapy yesterday, which I really think is mostly in the cure space, and I really wanted to give a bit of an update on where we are with HIV vaccine development as a prophylactic approach. So these are the sort of things I want to deal with in the next 30 minutes, which is why do we need a vaccine, where are we up to in that process, and what approaches are there in the pipeline for developing an effective prophylactic vaccine, both in terms of active immunisation and what we can learn from the role of passive immunisation and protection from the use of monoclonal antibodies with neutralising antibody activity. I want to talk briefly about some alternate approaches to vaccines, and then I wanted to just give you a quick update on where we're up to with late phase trials in this space. So why is a vaccine important? Well, there's that old saying that prevention is better than cure, and I think that very much holds true. And all other mechanisms of prevention require high-level recurrent compliance. So although PrEP and other biological interventions and behavioural interventions are very important, they all require prolonged compliance. Even when you're targeting particular populations, that is difficult to obtain, particularly when people are healthy. And vaccines, by contrast, only require short-term compliance. And that then converts into the fact that rather than giving an antibiotic or an antiviral on a regular basis, you're giving this as a one-off event, and the economics of that are compelling. Effective vaccines have eradicated infections, and despite the fact that we've had effective antibiotics, we're still not in a position, even with cheap antibiotics, to have come close to eradicating any infection with the use of antibiotics. So although this is a promising approach, and there is modelling to support this, in the real world we haven't managed to do it with any other infection, so why should we be able to do it with HIV? But the problem is that all the vaccines we have that are very effective, that work, really are effective against acute infections, and we're relatively poor at developing vaccines against chronic infections, particularly those that are intracellular. And I want you to think about those sorts of infections like malaria, TB and hepatitis C, where we have no vaccine. And there are clearly parallels with HIV. And one of our problems with developing a HIV vaccine is that we're not sure of what the correlates of protection are. There are very few people, apart from certain people with certain genetic profiles, that are resistant to this infection. And so really we're guessing at what the correlates are. And the experts believe that what we need is a combination of broadly neutralising antibodies and strong, to prevent the infection in the first place, and strong cytotoxic T cells to mop up any infected cells that get past these neutralising antibodies. As I said, natural resistance, except those people who have a CCR5 delta 32 mutation, is uncommon, and where it's present it appears to be transient. And a lot of the work we've done in trying to determine what these correlates are have been in people who are long-term non-progressors and elite controllers, and what slows down progression or prevents progression may not be the same sort of immune response as you need to prevent the infection in the first place. So, however, there are lessons that we've learnt from the large phase 2B and 3 clinical trials, from macaque models, and from naturally occurring neutralising antibodies, and I'm going to go through some of that data now. But just to make sure that people understand what we're talking about here, or what I'm trying to talk about here, is I want to define what neutralising antibodies are. So this is a schema of the virus getting into a cell, and what neutralising antibodies do is they bind to the envelope spike in such a way that they prevent the envelope spike interacting with the receptors on the surface of the cell that the virus is trying to infect. So it interferes directly with the process of viral entry into the cell by blocking the interaction, an effective interaction with CD4 or CCR5 or CXCR4. By contrast, binding antibodies that are effective at clearing the virus work a different way. They bind again to the envelope, but elicit responses from other cells in the immune system, such as NK cells or macrophages or monocytes, through their interactions with FC receptors or through their interaction with complement to induce phagocytosis and destruction of the viral particles that way, or killing of infected cells through a process called antibody-dependent cellular cytotoxicity, or ADCC for short. And so neutralizing antibody, this is a schema, a simplified schema of how the virus gets in, of the envelope trimer interacting with the receptors on the surface of the cell, and basically a neutralizing antibody will block one of these critical steps, either the binding of CD4 to the trimer, the interaction between the reconfigured trimer and CCR5, or the elicitation of the fusogenic machinery of the virus that fuses the viral membrane to the cellular membrane and allows the capsid to enter the cell. So neutralizing antibodies will act at one of these three sites. However, the envelope of the virus has evolved to be immunoavoidant, and it's done this in a series of ways. There's not just one mechanism by which it's immunoavoidant. The first thing is that it's highly mutable and it has these variable loops. The most important of these from a vaccine point of view are the V1, 2 and 3 loops, variable 1, variable 2 and variable 3 loops. And these areas of the virus are highly mutable. Their virus can mutate their amino acids in this place in these loops with impunity and escape or become resistant to an immune response. The interaction between CD4 and CCR5 is interesting as well, in that after binding to CD4, there is a cryptic face of the envelope that opens up that allows it to bind to CCR5. And that space is shown here. It's not going to... So in the upper right-hand corner, and you can see that there's that white space between the envelope protein and the cellular membrane. And that space is actually too small to allow an antibody to get in there. So even if you can make antibodies to this cryptic face of the virus, the antibody can't work. And in fact, none of the neutralizing antibodies that we've described act at this face, even though we can block that interaction with drugs like Maraviroc. There is this complicated trimeric structure of these three monomers associating. And then there is this thing called the glycan shield. So this is a glycosylated protein of these three proteins in association, and this protects the proteins. These glycosylation molecules protect the protein from the immune response. The immune response is very good at recognizing protein and foreign protein. It's not very good at looking at glucose molecules or their derivatives and generating an immune response. And in that lower right-hand corner, there is a model of the trimeric structure. And each of the protein monomers is shown in orange, light blue, and purple. And you can say to me, well, I can't see those. Well, neither can the immune system, because these proteins are covered in these blue balls. And those blue balls are the glycan molecules that are shielding the protein structure. So the immune system has trouble getting through those crevices to the protein that it needs to interact with. So the other thing is that although I told you that neutralizing antibodies and cytotoxic T cells are what we think is important, when we actually do the clinical trials, we get a different answer. So the only successful clinical trial we've done is the RV144, which was done in a large number of low-risk participants in this country. And it had a 31% efficacy. So it has borderline utility. but it had much higher efficacies in certain subgroups and in the first 12 months. And the results really make us think, because there was only low-level, limited breadth and transient neutralising antibodies produced by this vaccine. There were no cytotoxic T cells produced at all, and the T cells that were produced were cytotoxic CD4 T cells, and the antibodies, by and large, were binding only. So a lot of work by many groups in a coordinated effort have tried to pick this apart and understand what the correlates of immunity are in this clinical trial, to see if we can build a better vaccine based on the findings of this clinical trial. So what's been found out so far, I'm going to try and summarise in the next two slides. So antibodies to that first sequence of that initial part of the sequence of envelope, the constant region 1 and the V1 and V2 loops of envelope, appear to be very important. Binding to those, the correlates of immunity include binding to those regions, the breadth of the antibodies to those regions, and interestingly, these epitopes are not conformational, they're linear, so they're relatively easy to synthesize. And simple manipulation of this area of the envelope sequence can change the immunogenicity of this molecule. And these are monomeric molecules that were produced, so they're relatively easy to produce. So these are encouraging results. And these antibodies don't require some extensive somatic hypermutation. I'm going to be talking a lot about somatic hypermutation with the neutralizing antibodies, but this is what happens to antibodies as they mature over time to become more effective. And these, the antibodies that were produced do not need high levels of somatic hypermutation. But these antibodies were not broadly specific, and single amino acid changes abolished their ability to work. So simply one amino acid change in the V2 loop can abolish their efficacy. So the virus has great ease in escaping these responses. We produce different sorts of antibodies. They can be different isotypes. We have IgG, IgA, IgM, and interestingly, the ratio of the sorts of isotopes that are produced to this vaccine are very important. So we all think that IgA is important. It's part of the mucosal immune response, and you would think that IgA is important in protecting us from an infection that's mostly mucosally acquired. But in this instance, when you measure the IgA to envelope in the blood of these individuals, the higher their IgA response to the V1, V2 loop region, the less likely they were to have an effective immune response, a protective immune response. And this is because these IgA antibodies somehow block the interaction of the IgG antibodies with this region and stop the IgG antibodies inducing antibody-dependent cellular cytotoxicity. The most effective isotype produced seemed to be the IgG3 antibody, but these were very transient. So what we look like we need is to be able to produce high levels of IgG3 and lower levels of at least blood IgA. One of the limitations of this study is that only blood was taken. There was no studies done on the actual mucosal immune response. But certainly not all isotypes of antibodies are equally effective. So where are we with vaccine development? Which way should we go? Well, there are currently two schools of thought. One is to take those RV14 results, take the immunogens that were used, and improve the longevity and the immunogenicity of those responses to try and prolong the initial protective response that was most marked in the first year after the vaccination. And the other idea, the other camp is we try and induce or mimic the natural production of broadly potent neutralizing antibodies. And I'm going to spend the next part of the talk talking about neutralizing antibodies. So this phenomenon of being able to do single cell studies and characterize B cells and plasma cells at a single cell level have really revolutionized the discovery of neutralizing antibodies. A decade ago, we had 10 neutralizing antibodies that we'd fully characterized at a monoclonal antibody level. There were 4 of those 10 years ago. Now, as I'm going to show you, there are a plethora of these antibodies, and they're very important in understanding what we need to induce in terms of a protective vaccine immune response. And what we know from these studies is that these antibodies are rare. They take a long time. So most patients produce strain-specific antibodies, but only the rare patient produces broadly neutralizing antibodies. When they produce those, they produce them relatively late in the disease. These take at least a year after infection to be produced, and often much longer than that. And this is because of this need to accumulate a bunch of somatic hypermutations within the antibody. And this is driven by this complex evolution of the neutralizing antibody response. And this, in turn, depends on the initial immune response, the germline response, the initial envelope sequence the individual is exposed to, and then this sequential order of the immune response occurring to the envelope, driving out an escape mutation, a new immune response being generated to the new envelope, and so on and so forth over months and years to generate these high affinity antibodies that have broadly neutralizing antibody characteristics. And that clearly you're generating, you need good help during that time. So you need a coordinated response between your B and T cell response. And this is one demonstration of this. These are 2 structural diagrams with GP120 shown up the top. On the right, the GP120 is shown in green. The ribbon is the protein. The hatched area is the glycosylation molecules. And then the grey section down the bottom is the monoclonal antibody structure, or part of the monoclonal antibody. And what you can see is there's this knobbly finger protruding up past this glycan shield to interact with the GP120 molecule at an epitope. And so what this is required is this evolution of many mutations within the antibody to generate this knobbly finger that can find its way through the crevice of the glycan shield to interact with a particular part of the protein that then allows that antibody to be neutralizing and block entry. Neutralizing antibody affinity and therefore its effectiveness and potency increases with increasing mutation. And I just want to show you on this graph that to get high affinity, you need 65 mutations. The traditional antibodies that we generate to TET-tox, which is a really effective vaccine, have 3, maybe 5 mutations. This is 65 mutations that we need to generate these sorts of antibodies. This is a real challenge. So we're learning a lot about these antibodies because we're producing a lot of these antibodies. There has been a really very big effort to chase these antibodies down with various techniques which are outlined on the right-hand side of that table. But you can see there's no longer just 4 of these. There's 20 and there 20 or more, and there's a growing list of these, and these are being studied intensively by groups around the world. So these are still rare. They're hard to find. You need heroic efforts to find them within an individual, and not all individuals produce them. So you have to screen tens of thousands of B cells to find one. No one technique finds any of these monoclonal antibodies. You need to use several different techniques to find all of them. What we still know is that these broadly neutralizing antibodies develop over many months to years, and they result from this coevolution that I talked about between the virus and the host, and this sequential escape from the immune response. And interestingly, some of these antibodies, if you mutate these antibodies back, if you get the actual sequence that codes the antibodies and you mutate them back to the germline, they don't bind to HIV at all. So they've been produced originally to another pathogen, and then the immune response then cross-reacts with HIV. So you can imagine that if that's what you've got to do, developing a vaccine to mimic this response is not going to be a simple thing. But the other thing we know is that these antibodies mostly bind to one of 5 sites, which is shown on this diagram here. So in this diagram here, it's the trimeric structure of the envelope. The light grey is GP120, the dark grey is GP41, and then the coloured areas are the areas where the families of monoclonal antibodies bind. And so there is, in purple at the top there is the high mannose, what's now called the high mannose region, which is sort of like the old V3 loop specific antibodies. Then next door to that is the apex area, what's called the apex area, where antibodies that we think about binding to the V2 loop region bind. In green, antibodies that is the site where antibodies that block the CD4 binding bind to this molecule. In red is this area called the GP120, GP41 interface, and then right down the bottom there, where the membrane is sitting, is the membrane, in yellow is the membrane proximal region, epitope region, where another family of neutralizing antibodies bind. So almost all these antibodies bind to one of these 5 regions. So these seem to be the 5 important neutralizing domains of this complex molecule. So what have we learnt? We know that these are rare in natural infection, and therefore they're going to be difficult to induce by vaccinology. If they're not common in nature, they're going to be hard to mimic. You need chronic antigen stimulation, you need evolution of the immune response, you need cooperation with follicular T helper cells, because that's what drives somatic hypermutation in the B cells. And so the vaccine strategy needs to control both evolution of the host response and correct pairing of the heavy and light chains. And I can tell you that we don't have any vaccine that we're using at the moment that comes close to doing this. There are, however, models where people are trying to mimic this in both macaques and in mice, and one of these examples is shown here, where you sequentially immunise with different envelopes to try and mimic the natural response of the evolution of the envelope, and therefore develop higher affinity antibodies. So there are no immunogens that currently induce neutralizing antibodies in vivo, and there are no immunogens ready for this sort of approach for late-stage clinical trials. And if you need chronic expression of these antibodies, often you need to give these with a vector that is chronically expressed, and retroviral delivery or CMV vectors have their own safety issues. At the same time as generating the B cell response, you need a coordinated T cell response, and this needs to be happening in the tissues, in the lymph nodes, in the follicular T helper cells. And we need to learn how to be better at inducing those responses. And we do have some hints to that from the attenuated models in macaques, and also the recombinant CMV vaccine that's being looked at in macaques. So these antibodies can be used, they can be produced at high volume and infused into muscles, into macaques, and can be used to protect macaques from challenge. And this is a list of the clinical trials that have done that. And if you look at that table clearly, you can see that some of these are very effective with complete protection of those macaques from SIV challenge. So you can use, so in vivo, if you can induce these antibodies, they look like they're going to be effective, because when they're used as passive immunisation, they work. We do have to be a little cautious. The shivs that are used for this are not HIV, macaques are not humans, and so what we're doing in the laboratory may not be mimicked in humans. When you infuse these monoclonal antibodies, you want them to go to the right places, you want them to go to the areas where you're going to get viral challenge, and by and large, this is what seems to happen when you do these sorts of studies in macaques. And there are currently studies in humans, one is shown here, but there are others that are looking at the use of passive immunisation with these monoclonal antibodies or combinations of these monoclonal antibodies. And one of the, apart from looking at protection, these are also looking at biodistribution. And there are a bunch of these antibodies that are in the pipeline, and some of these are listed here. And people are looking at monoclonal antibodies to each of those binding sites and epitopes that I showed you in that diagram a couple of slides ago. But people are also trying to engineer antibodies. So if you use one of these antibodies at a time, the virus is likely to escape, so you need to use combinations. Another way of mimicking combinations of antibodies is to actually engineer antibodies with bi or tri specificity, so they're binding to more than one epitope at the same time, and so you have a more potent antibody that is less likely, the virus is less likely to be able to escape from, because you're targeting two or three positions in the virus in the same way as with combination antiretroviral therapy, we target the virus at multiple sites. The limitations of passive immunisation as a preventative strategy are significant. You need these combinations for broad activity. They have relatively short half-lives, even when engineered to have longer half-lives. And so you need to redose these things all the time. A one-off treatment is only going to last several months at best. They need to be given parenterally, often IV, and they're expensive both in production and then in the maintenance of the immune response. So one way to get around at least the short half-life of these things is to engineer a molecule like this into a vector, a gene therapy vector, and deliver that. And one example is given here, where a tri-specific molecule that binds to CD4, binds and blocks the CD4 binding, that blocks the CCR5 binding and is effective in protecting macaques, can be delivered as gene therapy with an adeno-associated virus vector. So where are we up to with clinical trials? So the clinical trials, the late phase clinical trials, are really around variations on the RV144 theme, and various immunogens developed from what we've learnt from the RV144 trial have been through phase 1 and phase 2 testing, and have in South Africa recently entered phase 2-3 trials. And there are two of these trials that have started in the last couple of months. One is HVN702, which is a modified RV144 type immunisation regime, where there are more vaccines given over a year. There's a new adjuvant that hopefully generates better immune responses. And instead of being an AE-B stroke B sequence, it's a subtype C sequence to mimic the population. And the other trial that's just started takes a different approach, where a mosaic vaccine that hopefully has broader strain specificity is delivered as a combination of an adeno-26 vector with a GP140 protein boost. And as I said, both of these trials have just commenced in South Africa, and we'll have the results of those in the next 3 to 5 years. So in conclusion, there are two almost antithetical approaches to vaccine development at the moment. One is an empiric approach that is looking at the success of the one success of the clinical trial we've had, the RV144, and modulating the immune responses that were induced, or modulating the immunogens that induce that immune response. And then there is what is called a more rational approach, but will take much longer to develop, of trying to understand what a neutralizing antibody looks like and then trying to induce those with a vaccine. I'll leave it there. Thank you.